Good morning, everyone. I'm Kathyana, and for my final year research project, I determined antioxidative properties of three selected cyanobacteria as Croococcidiopsis species, Merismopedia species, and Gleocapsa species isolated from water bodies in Sri Lanka. Before proceeding into my presentation, if I ask you whether you are familiar with the word cyanobacteria or not, some of you may answer it as yes, while some of you won't. But if I showed you this picture and asked you whether this is a common scenario to you or not, then most of you will answer it as yes. This is the most common form of cyanobacteria we encounter in our day-to-day life in our surroundings. So cyanobacteria are a very primitive group of organisms, and they are also known as blue-green algae due to their unique color. Characteristics similar to both algae and bacteria are shown by these cyanobacteria, and they have the ability to thrive in any harsh environment. We can see them as unicellular, colonial, or multicellular filamentous form. In the present world, cyanobacteria are being utilized in many industries. In the food industry, they are being used as a food supplement, a color enhancer, and even NASA is experimenting on cyanobacteria as a food booster for their astronauts. In the pharmaceutical industry, cyanobacteria are being utilized as anti-cancer, antiviral, and antibacterial agents. Most of these extracts are still in the clinical stage. In the cosmetic industry, cyanobacteria are being utilized in the production of sunscreen, moisturizing cream, and in the production of deodorants as an odor mask. Now let's focus into the second keyword of my research project, which is antioxidant. Reactive oxygen species or ROS are produced within our body as a response to the external and internal stresses. Continuous production of these reactive oxygen species for a longer time cause oxidative stress. Due to this oxidative stress, diseases like cardiovascular disease and cancers can be happen, and it even speed up the aging process. To fight against this oxidative stress, there comes the antioxidants. So now you may get the question, why cyanobacteria as an antioxidant source, even though there are so many other sources available in the world? The most common natural antioxidant sources available are berries and beans. But these are not a feasible and viable option in the industrial production because of the high raw material cost and the longer time they take for the mass production. Synthetic antioxidants like BHT and BHA are once again not a feasible option because they have been found out to be causing gastrointestinal tract problems, skin allergies, and even causing cancers. So there's a high need of a new natural source of antioxidant which can be utilized in the industrial level. There comes the cyanobacteria with a very high growth rate. So the objective of my study was to determine the antioxidative properties of selected cyanobacteria, Croococcidiopsis species, Merismopedia species, Gleocapsa species isolated from water bodies of Sri Lanka. The place I carried out my research was the Nutritional Biochemistry Laboratory at the National Institute of Fundamental Studies, Hantana. And these are the original sources which the National Institute of Fundamental Studies had collected these three species of cyanobacteria. The places are Kurunagala Tank, Wellwater of Girandurakote, and Hot Spring of Mahapalasa respectively. The pure cultures were provided and I inoculated them in a BG11 media. In the yellow colored cages, you can see the subsequent growth I observed within the first few weeks after inoculating. Then subculturing was done, and in the last cage, you can see how they are thriving under an artificial light source and oxygen source, which was provided through an electric air pump. In every once a week, morphological identification and contamination was observed under a light microscope. As you can see in the red color box, there was a contamination in our tank culture which led them to eliminate the tank culture from our experiment. After 1.5 months, we harvested them, centrifuged them, freeze-dried in order to remove the excess water and obtain the dry cell mass. 30 milligram of dry cell mass was added with 1 milliliter of water and 1 milliliter of 1:2 volume by volume methanolic solution. The reason for using two solvent was that in some of the industrial applications like pharmaceutical industry, they does not consider alcoholic solvent as safe or GRAS generally recognized as safe. So in order to examine the possibility of using these antioxidant in those kind of industries, we had to use the water as a solvent also. So the solvents and the dry cell mass were sonicated in order to break down the cell and take out the intracellular things out. And the cell-free extract was pipetted out and subjected to 2 phytochemical assays and 3 antioxidant assays, as you can see in the slide. The full form of the abbreviations are also can be seen in the slide, but for the convenience, I will use this abbreviation to discuss about my findings. Statistical analysis was done using IBM SPSS statistical software, and one-way ANOVA was used. Mean comparison was done using Duncan's test at 95% confidence interval. This is the dry cell mass I obtained from the 3 separate species of cyanobacteria. You can see that there is a higher dry cell mass from the Croococcidiopsis species than the other 2 species. When I refer the past studies and the articles, I found out that Croococcidiopsis species is being considered as a possible candidate to grow in the Mars due to its ability to survive in the harsh environments. This justifies the higher growth rate and the high dry matter yield we took from the Croococcidiopsis species in our study. Then phytochemical assays was done. Before straightly going into the antioxidant assay, phytochemical was analyzed in order to take an idea about the antioxidant capacity of these 3 species. The phenolic content was analyzed using Pauling Chroococcidiopsis method. The Pauling Chroococcidiopsis reacted with the phenolic available in the sample and turned the color into colorless form into the bluish color. The intensity of the bluish color is measured through a microplate reader and compared with the gallic acid standard curve. Gallic acid was used because gallic acid has been found out to be one of the phenol most available in the cyanobacteria. The highest phenols was observed in the water extract of Croococcidiopsis, while the lowest was observed in the water extract of the Merismopedia species. Flavonoid content also measured. The flavonoid are also a polyphenol and has a very good antioxidant activity. In here, we use aluminium chloride method. Flavonoids available in the samples make a complex with the aluminium chloride and give out a reddish color. The reddish color is measured through a microplate reader and compared with the catechin standard curve. The catechin was used once again because catechin has been found out to be a flavonoid which is abundantly available in the cyanobacteria. The highest flavonoid was observed in the methanol extract of the Merismopedia, while the lowest was observed in the water extract of the Gleocapsa species. Now let's move to the antioxidant acid. The first one was the ABTS antioxidant acid. In here, ABTS radicals get neutralized from the antioxidant available in the sample and turn its original greenish color into a colorless form. This intensity is measured from a microplate reader and compared with the Trolloc standard curve. The highest ABTS antioxidant activity was observed from the methanol extract of the Croococcidiopsis species, while the lowest was observed from the water extract of the Gleocapsa species. DPPH antioxidant activity measured somewhat differently than the other 2 antioxidant assays. In here, IC50 value is measured. IC50 value means the concentration required from a sample to inhibit 50% of the radicals available in the DPPH. So the higher the IC50 value, the lower the antioxidant activity. In here, the highest antioxidant activity was observed in the methanolic extract of Merismopedia, while the lowest was observed in the water extract of the Gleocapsa species. Finally, ORAC assay was done. In here, we used a Trolloc standard curve in order to compare the ORAC values. The highest ORAC values was shown by the methanol extract of Merismopedia, while the lowest was shown by the water extract of Gleocapsa species. So in order to determine the relationship between these phytochemical assays and antioxidant assays, we measure the correlation between them. For the phenolic content, there was a positive correlation with ABTS, while there was a negative correlation with DPPH and ORAC assays. For the flavonoid content, there was a positive correlation of ABTS and ORAC assay, while there was a negative correlation with DPPH antioxidant assay. So, by concluding my final year research work, I would like to emphasize that these three species of cyanobacteria can be utilized in the industrial production of antioxidant in many industrial applications like cosmetics, pharmaceuticals, and foods, with further modification in culturing and extraction procedures. And within these three species, Merismopedia highlighted itself as a very good source of antioxidant than the other two species. Thank you.